I think it's going. All right, so we're going to talk about the nervous system and neurons. And I got to get my pen. Okay. All right. There we go. I hate this thing. I like to... Hang on. There we go. All right, so I very rarely tell you guys to memorize anything, but these next two slides, I do want you to memorize. So let's start with this one. This is something I've already done on the board for you guys in class, okay? And so it should be a lot of review, and then we'll talk about the next slide, which again is something I want you to memorize. I very rarely say that, but I'm going to say it now. The way the nervous system works is basically the nervous system is composed of information coming in, or input, from your sensory receptors. It can be external, like vision, or hearing, or pain, or touch, hot, cold, anything like that. Any kind of external sensory perception, use your ears, your nose, your eyes, your fingers, anything like that. There's also internal receptors, things that are monitoring things like blood pressure, oxygen levels, carbon dioxide levels, sugar levels. All these kind of things are constantly being monitored by your body. All of that is information. We call that input, or sensory input. So for example, here you've got the eye. It sees water, and it sends that signal to the brain. Okay? Now, the brain and the spinal cord are basically your computer. That's where the integration happens. Actually, it says that right there. Integration. So this is your computer. It gets the information, and it makes the decision. All right, what is that, and what needs to be done about it? All right, so then once that decision has been made, you send out output, which is basically the response. Now, this response could be a motor response, like moving a muscle. It could be a response to the heart to increase beating rate. It could be a response to the diaphragm to increase your respiration rate. It could be a response to your pancreas if your sugar levels are changing to increase or decrease the amount of insulin you're making. Okay, it could be any kind of response to your internal organs to make some kind of change. Okay, so it could be an external change, like to move a muscle, or an internal change to regulate some kind of homeostasis. We're trying to maintain homeostasis. That's the whole that's not right. Homeostasis. The whole idea is to keep our body regulated at the same heart rate, same oxygen levels, same carbon dioxide levels, same sugar levels, same pH. We want to keep all this balanced. So we're constantly getting feedback from our external and internal environment. Now this is the slide. I know it looks busy. It's huge. It's got lots of information. But we're going to break this slide down bit by bit by bit over the next 3 weeks. Okay? So this is kind of, I put it all together for you, and then we're going to break it down as we go along. But this is basically, if you can memorize this, you'll know a lot. All right? When we talk about the nervous system, we break it into two main divisions: the central nervous system and the peripheral nervous system. And I'm going to call it CNS and PNS. Okay? So if I don't say central or peripheral, I'll probably say CNS or PNS. The central nervous system is the brain and the spinal cord. That's it. It's centrally located, okay? It's the central nervous system. It's the computer. It's where the integration comes. That's where the decisions are made, okay? There are decisions made by your spinal cord. We have things called reflexes, where it doesn't even need your brain to even make a decision. Your spinal cord kind of makes that decision for you. We have the peripheral nervous system, or the PNS, and that are the cranial nerves, the spinal nerves, and that's basically what's sending information out to the body and then bringing information back in from the body. All right, so somebody yesterday said something about peripheral vision. What does peripheral vision mean? You're looking to the what? To the side? Yeah, so the peripheral nervous system are the nerves out to the side. Okay, so they either come off the cranial, the brain, the cranial nerves, or they come off the spinal cord, and they're called spinal nerves, and we're going to talk about that in lab. Now, when we talk about the peripheral nervous system, we've got to divide it again. We've got to divide it into two divisions. You have an afferent and an efferent division. And you'll hear some people will say afferent and efferent. It's kind of like adduction and abduction. It kind of sounds the same. So it is perfectly acceptable to say afferent and efferent, and that way people know what you're talking about. So don't feel like you're a hick or anything saying it goofy. Everybody says it that way. Afferent and efferent, so you know what you're actually saying. Afferent is the sensory division. It's bringing information in, okay, back to the central nervous system. The efferent is the motor division, and it's taking out the response. So the sensory is the stimulation, whatever was stimulated, whatever sensory receptor was stimulated, is bringing that information in. The efferent division is taking the answer or the response back out. Now this efferent division can go to muscles or it can go to glands. So you divide the efferent into two divisions, 1 and 2. We have a somatic nervous system and an autonomic. What's autonomic sound like? Automatic, exactly. It says automatic. So somatic is voluntary. So just think about muscles. Our skeletal muscles are voluntary. So your somatic nervous system is when you're in control, like when you're moving a skeletal muscle. The autonomic is your visceral or involuntary. Your viscera are your organs, right? So you don't involuntarily control your organs. That's involuntary. So your autonomic nervous system sends a response to your viscera or your organs, things like cardiac muscle, smooth muscle, glands, endocrine glands, adrenal glands, pancreas, gallbladder, any of the glands. You're going to talk about this a lot in 202. This is basically what 202 is about. How the glands, the organs, the heart, the lungs, digestive system, reproductive system, urinary system, endocrine system, how all those work. And that's part of your autonomic nervous system. Now, your autonomic nervous system is also divided. Okay, you divide it one more time into sympathetic and parasympathetic. And you've probably heard of these words before, maybe not. When we talk about sympathetic, you may have heard the words fight or flight. Parasympathetic, we usually use the terms rest and digest. And what that means is there's two divisions of the autonomic nervous system that are in control in different situations. So say, for example, you're out on the street and a bus falls on your best friend, okay? Which nervous system do you think is going to take over, rest and digest or fight or flight? Fight or flight. When you hear people say, oh, they got an adrenaline rush and they were able to do superhuman things, that's your sympathetic nervous system. It's increasing your heart rate, your respiration rate, making you more powerful. That's kind of your fight or flight. If a tiger's running at you, I guarantee your sympathetic nervous system is going to kick in and get you out of there. If your parasympathetic nervous system kicks in, then you're dinner. Okay, so that's you want this division of being able to get you out of a situation, to make you stronger, make you more powerful, whatever you need, and then there's just your normal everyday rest and digest. And we're going to talk about these in detail as we move along. What you can notice on this slide too is you've got sensory fibers, okay? So this is your afferent division. You'll notice there's somatic fibers, so from your skin, for example, temperature, pain, touch, things like that, anything that's telling you something about the outside environment. You have visceral fibers. This is one coming from the stomach. That would be something, when would you have a visceral fiber, maybe from the stomach, be stimulated? What might cause that nerve to fire and be active? Do what? If you're hungry, yeah. If you're hungry, you start getting what's called borborygmus, where your stomach, that's like growling. There's actually a name for that when your stomach growls. And so when it's empty and the acids are kind of building up, there's a signal that goes to your brain. It goes, hey, I need some food down here. Or say you just had Thanksgiving or Easter dinner and you're just stuffed, you're full. There's actually some pain receptors Okay, your stomach's stretched, telling you, okay, we're good, no more food down here. Okay, so you've got visceral receptors coming from your organs. Then you've got responses. You've got nerves that go back out from the spinal cord, either to skeletal muscle, those are somatic, or to various organs, either to the heart or the bladder, pancreas. Like I said, we're going to do these more and more detail. But take this picture and really try to study that, because there's a lot more information on this picture than you realize. As we go along, you'll be able to go back to this picture and kind of start putting things together. Yeah. It's figure 11-2. 293? 387? 387. When we talk about the nervous system, we talk about tissues, and I will go ahead and tell you this, I'm not going to make you look at slides this time for the lab. If you want to see a nervous tissue, we will, but if you don't, then it's up to you. I'll have some slides out, but I'm not going to make you responsible for it for the practical. But when we talk about nervous tissue, there's basically two main cell types. You have neurons, and that's the main cell, okay? It's the excitable cell. It's the thing that actually sends off an action potential, does the signal. It's the one that does the work, okay? That's like when we've talked about neurons before, the one that goes to the neuromuscular junction, you got neurons in your brain. So that's the main type of cell. Well, we've also got supporting cells. They're called neuroglial cells or glial cells. And these are support cells, things that kind of help hold the brain together, hold the spinal cord together, all right? There's 5 different types of support cells. You have astrocytes. I've got a picture of each one, so we'll go through these quickly. Microglia, ependymal cells. Some people say ependymal. I say ependymal. Helps me spell it better. Oligodendrocytes and Schwann cells. The astrocytes are your scaffolding, your support, okay? Your microglia or microglia are your nurse cells. Your ependymal cells are lining. They line cavities. I'm going to show you some more information on this, so hang on. Then you have 2 types of cells that make myelin. So let's go to each one of these and I'll talk about them. Your astrocytes are the most abundant. There's lots of them, lots of branching. So you can kind of see they look like scaffolding. They kind of hold everything together. They're almost like connective tissue, kind of like the glue that holds everything together. They're support and brace. They do play some role. They do have some role in information processing because they do connect to different neurons. There's some thought that they do actually help in information processing, but we won't go into a whole lot of detail on that. The name, astrocyte, that comes from cyte means cell, astro means star, so it's kind of a star-shaped cell. Microglia, or microglia, it helps you spell it better, are basically nurse cells. They're associated in the nervous system. They migrate in, and they will phagocytize, which means eat, okay, bacteria, viruses, anything, blood cells, anything that's any trash, debris that needs to be gotten rid of, they're going to take care of it. So they're your housekeeping or nurse cells. The ependymal cells or ependymal cells, these are the lining cells. And inside your brain, you have cavities. These cavities are called ventricles. Just like the ventricles in your heart. The ventricles in your heart are just spaces or cavities. So you have cavities inside your brain that's filled with fluid. That fluid's called cerebrospinal fluid. Okay? That cerebrospinal fluid basically is in and around, it flows. Here's your brain. Cerebrospinal fluid flows all around the brain and into the brain and down the spinal cord. It's kind of everywhere. The brain basically floats in cerebrospinal fluid. But there's no pump. There's no heart. There's nothing to move that cerebrospinal fluid. And it needs to move around because it's got, nutrients in it and oxygen and things like that. It's got to be able to move. So these ependymal cells have cilia, which can move, and will actually help move that and let that CSF, cerebrospinal fluid, CSF, flow through those cavities. Okay, so that's their job, to kind of keep that cerebrospinal fluid going. Now, you have two cells that make myelin. You have oligodendrocytes and Schwann cells. Oligodendrocytes are in the CNS, brain and spinal cord. Schwann cells are in the PNS, okay? They're very different cells, and they work a little bit differently. What I want you to notice, this is an oligodendrocyte. It's one cell, and it has branchings that go out and wrap adjacent nerve fibers with myelin. Remember, myelin is just a fatty substance used for insulation. So in the CNS, you have one cell that branches out to cover adjacent neurons with myelin. Schwann cells are different. And sometimes you'll see the word neurolimocytes. I put it in there because some people call them that, but Schwann cells that's what I call them, and that's what I'll test you with. They're found in the PNS, the peripheral nervous system, cranial nerves, spinal nerves, all the nerves that go out to your arms and legs. And what you'll see is when you look at an axon, each one of these is a Schwann cell. So there's multiple cells on an axon. So each one of these is a Schwann cell, and basically it wraps itself around the axon, insulating it. Sorry. All right. So some special characteristics of nerve cells. Your nerve cells are long-lived. Okay, so if you have a nerve that goes from your spinal cord all the way down to, say, your little toe, you've had that since you were born. Okay, and it stays with you all your life. So they're not real mitotic. They're amitonic. They don't divide. They don't reproduce. They don't replace themselves like every other cell in our body. Our muscle cells are constantly growing and dividing and repairing themselves. Nerve cells, once you've got them, and they live a long, long time. Now, I think I'm going to say this in just a second, but I'll say it now too. In the CNS, there's very little or no repair of neurons. In the PNS, there is some repair. And I think I'll talk about this again. But think about if you were to cut your hand, and you might cut a nerve, okay? It will actually, the skin will grow back, the vessels will grow back, and those nerves will basically reconnect. It'll repair itself, so you don't have a dead feeling forever and ever and ever. You're going to get that feeling back in that area. Your peripheral nervous system will heal itself. Your central nervous system, very little repair or maintenance or regeneration occurs. So if you have severed the spinal cord, mm-hmm. Mm-hmm. Yeah. Yeah, I have a dead spot right here because when they cut the nerve doing my neck surgery, it didn't quite grow back, so I actually do have a dead spot. Depends on what kind of damage. Was it severed or was it nicked, you know? And so, like, I have a dead spot too. So they can grow back. They don't necessarily grow back. Or they don't grow back right. You don't have the same feeling you did before. But in the CNS, there's no repair at all. So if you sever your spinal cord, it's severed. It's not going to grow back. So that's one of the main differences. You do get some repair in the PNS, very little or none in the CNS. If you have brain damage, it's very hard to get that part of the brain to come back. Those cells are damaged. It can. You can be retaught to do things. We'll talk about lateralization. We'll talk about different parts of your brain, left and right. Sometimes you can relearn things, but not always. Depends on what it is. Physical things you can often learn, memory, things like that it doesn't come back. Nerve cells are highly metabolic. Okay, that means they generate a lot of ATP. They use a lot of oxygen and glucose. If you've ever, especially if you've been on a diet or you skip breakfast or you skip lunch and you get that headache, you're like, oh man, and so what do? You go reach for a candy bar or a Coke or something. You're looking for fast sugar. That headache is a signal to your brain, hey, we're running out of energy up here. We need some sugar, because your brain is constantly using oxygen and glucose to do cellular respiration to make ATP, okay, so that it can function. That's one of the reasons why if you ever have more than 3 to 5 minutes without oxygen, you're looking at having brain damage. Stroke victims, heart attack victims, drowning victims. The first thing you want to do is try to get oxygen going to the brain, because the longer the brain cells or neurons are without oxygen, the more likely they are to die, and then they don't come back. So they're really metabolic. You need lots of oxygen, lots of glucose to keep these things happy. Their membranes are electrical. That means they can generate action potentials. And unlike when we've talked about a nerve going to a muscle, we've talked about a neuromuscular junction, that's one nerve going out to a muscle. In the brain and in the spinal cord, neurons can actually interact with each other. Okay, so what we've talked about so far is having a neuron going out and innervating a muscle. In the brain and spinal cord, you'll have neurons that interact with each other. They're signaling each other. And this is how we make our conscious thought. This is how we make memories. Cells will interact with each other. It's how if you smell a rose, you may think of your grandmother, or if you smell chocolate, you may think of Valentine's Day. Just some kind of, that's how we build our memories, are these neurons being able to interact with each other. And we'll talk more about that as we go along as well. All right, this should be total review, so I'm just going to fly through this real quick. This is a motor neuron. We have one in the lab. We're going to go over it in lab. You'll be tested on that as part of your practical. But you've got your cell body, also called the soma. So here's your cell body. You have the nucleus. This little nucleolus inside the nucleus. You have dendrites. These are the receptive ends. That's where the information is coming into the cell. You have the axon hillock. That's where the action potential is generated. Action potential or impulse travels down the axon, goes to the axon terminals. That's the secretory region. What's in the axon terminals? Neurotransmitters. Could be acetylcholine, could be something else. Depends on what kind of neuron it is. The axon is wrapped with Schwann cells. And between each Schwann cell is a gap called a node of Ranvier. So that's review from what we did when we talked about the muscle. Now, what's going on in each part of the cell? The soma, or you'll see the word perikaryon. That means around the nucleus, basically what cell body. So you may see the word perikaryon. I like the word soma better, because soma means body. So it's the cell body. It's the biosynthetic region, just like any other cell. That's where the nucleus is, making proteins, things like that. You've got a well-developed Golgi apparatus. That's for protein packaging. You've got a rough ER, a rough endoplasmic reticulum. You've got something called Nissl bodies, and when we get into lab, I'll show them to you on the model, and I will refer to them as the candy sprinkles. Okay, and you'll know what I'm talking about, so you'll remember which one the Nissl bodies are. They're chromatophilic. Chromo means color, philic means love, so they're color-loving, so they're kind of colorful. You have things called neurofilaments. If you look at a neuron, remember, like a motor neuron, it could go from your spinal cord all the way down to your little toe. That's a very, very long neuron, okay? There's got to be a way to get sugar, proteins, oxygen, waste, whatever, back and forth down this really, really long cell. So I like to think of the neurofilaments as train tracks, okay, or a transportation system. It's a way to move stuff up and down the neuron. So that's what they're for. You have your axon hillock. Again, that's where you make the action potential. And when we talk about the central nervous system and peripheral nervous system, there are parts of the central nervous system where you have lots of cell bodies, and their axons kind of come out from it. Same thing in the PNS, where all the cell bodies are kind of clumped together, and then their axons kind of come out from there. If you're in the CNS, that's called a nuclei. It's a bundle of cell bodies. And if you're in the PNS, it's called a ganglia. And so we're going to talk about some ganglia and nuclei as we go along and talk about different parts of the body. So if I use the word ganglia or I use the word nuclei, you'll know that's just a grouping of cell bodies either in the central nervous system or the peripheral nervous system. Okay, you have your processes. You have the dendrites, which is the receiving end, the axons, which is the sending end. Now, in the CNS, you have your brain. Let's do a cross-section of the brain. I'm a horrible artist, y'all. That's the brain. If there's a region of the brain that works together, say there's a bundle of neurons that all work together kind of for a common goal, that's called a tract. Okay? In your spinal cord, there may be tracts of information that maybe all come from the left arm, for example. All that information goes together. That's a tract. In the peripheral nervous system, as things go out or come in, they're called nerves. So you don't have nerves in your central nervous system. You have tracts. You have nerves in your peripheral nervous system. They look kind of the same, but just kind of a different terminology. Let's see, dendrites, they're the receptive input area, and they are electrical. They will, here's your cell body, here's your dendrite. So you have an action potential coming across from maybe another neuron. It will synapse on this dendrite, generate an action potential here. That action potential will be carried to the cell body, regenerated at the axon hillock, and carried down to the axon terminal. So they can receive and generate or basically transfer electrical energy to the cell. Again, this should be hopefully a lot of review for you guys. Axon, again, that's the impulse conducting region. Takes information away from the cell body to the the axon terminals. These axon terminals, sometimes you'll see them called synaptic photons. So you may see that term. And basically that's just those axon terminals where you've got those vesicles that contain the neurotransmitters. And it could be acetylcholine, it could be epinephrine, it could be dopamine, serotonin. We're going to talk about all the different neurotransmitters, but they're all stored inside these axon terminals. All right, why do we have myelin? Myelin is basically a combination of proteins and lipids. Lipids are fat. Fat does what to our bodies? Why do walruses have a lot of it? Insulation. So fat insulates these neurons, okay, makes their, speeds up their impulse transmission, makes them faster. So we have myelin wrapped around the axons to speed them up, make them fast. All right. In the PNS, again, we have Schwann cells. I'm going to show you a picture of this. When you think of the Schwann cells, the way I like to think about it, imagine a biscuit dough or pizza dough, okay, and put a marble on it. All right, and the marble is the nucleus. Now spread that out really thin. So now you've rolled it and rolled it and rolled it, and you kind of push the nucleus is kind of in real thin or sticking out of that thin dough. Now take a hot dog, okay, and do a pig in a blanket. Roll that biscuit dough or pizza dough on that hot dog. As you do that, that pushes the nucleus to the outside. It winds up on the outside. So here we've got our biscuit dough, and we're wrapping it around this axon. And as we do that, the nucleus keeps getting pushed to the outside. So the outside layer is called the neurolemma. That's the layer that basically winds up with the nucleus in it. So the outside layer is the neurolemma, and a Schwann cell basically just wraps itself around an axon like a pig in a blanket. That's the easiest way to think about it. Oligodendrocytes kind of work the same way, except the nucleus stays in that central area, that central part of the cell, and it sends its processes out and then wraps around the axon, again, like a pig in a blanket. But it goes to, one cell goes to several neurons instead of a Schwann cell, where the axon's got a Schwann cell here, a Schwann cell here, a Schwann cell here. A little bit different way of working. Okay, gray matter and white matter. What does that mean? Anywhere you have white matter, and if you've worked on that case study that's due to the blackboard, you've probably seen a little bit of this information already. White matter is anywhere you've got myelinated fibers. Okay, so that's basically axons. Gray matter is where the cell bodies are accumulated. There's no myelin there, so there's no white color. Myelin is fat, and so it looks white. So anywhere you see white matter, you have myelinated fibers. Anywhere you see gray matter, unmyelinated. So let me draw a picture of a spinal cord. This is a spinal cord. And it basically has a butterfly-shaped central core to it. It is gray. It's dark. When we look at a spinal cord, when we do the sheep brain, you're going to notice it's dark in color. It's the gray matter. Everything around it is the white matter. So what's happening is all your little cell bodies are contained in this gray matter, and they send out their axons through this part of the spinal cord, and that gives you that white color. So gray matter and white matter. Now we talk about neurons. We have talked about this same structure over and over and over. Dendrites on this side, a cell body, an axon terminals. There are different types of neurons. There's basically 3 different types based on their structure. They don't all look like that. We have multipolar, bipolar, and unipolar. Let me go to the picture. Multipolar means there is more than 2. So more than two processes on the cell body. Okay, so in this case, there's always one axon. One axon, you've got 2, almost 3 dendrites that then divide even further. On this one, you have one axon, 1, 2, 3, 4, 5, 6 dendrites. So there's more than 2 somethings connected to the cell body. That's multipolar. These are the most abundant. You find them just about everywhere. If you have bipolar, that means you have 2 processes. Coming off the cell body. So you have always one axon, and there's just one dendrite coming off the cell body. So you just have 2 things. Bi means 2. If it's unipolar, that means there's just one process coming off the cell body. It then divides into an axon-like and dendrite-like area. So here's one process, goes into the axon, and then it divides kind of into a receptive dendrite type neuron process. These are specialized cells. You're going to find these bipolar, particularly where you have sensory organs in your smell, your olfactory, the ear, the eye, any of your special senses. A lot of times are bipolar neurons. Your unipolar or even mostly in the PNS, and they're found in the dorsal root ganglia. Remember, ganglia is basically a grouping of cell bodies in the peripheral nervous system, and I'm going to show you one of those in lab. So you have different types of neurons based on their structure, multipolar, bipolar, or unipolar. You can also classify them based on what they do, their function, okay? So we've already talked about the different types of neurons. Some neurons bring information in. They're sensory or afferent. They bring information to the central nervous system. Some neurons take information away. They're efferent, so they carry it from the CNS out to the gland or organ or muscle that it's going to respond. And then in between the sensory and the motor, you have the interneurons. And this is your brain and spinal cord. This is where the decision's made. This is a computer. So some neurons are purely sensory, some are motor, and then some make decisions. There is a video by Ms. Graham on spinal reflexes. It's very good. I suggest you watch. I think that's even one I've got a video quiz on. Make sure you watch that one. She does a good job talking about the different types of neurons and reflexes. Again, it's just a picture. You can see you've got motor neurons taking information out, sensory neurons taking information in, and then inside the spinal cord, you've got some motor neurons sending information. Whoops, sending information. Let me do that again. Where's my eraser? What would happen? Wait a minute. Yeah, you know why these things get crazy on the videos. So what would happen is, on the spinal cord, you would have information coming in, okay, sensory information coming in, a sensory neuron. You have an interneuron in the spinal cord to make the decision, and then it would send a response back out. So for this example, it might be that you're cold. So you have a sensory impulse telling you that the skin's cold, your body temperature's dropping, you're getting cold. You want to start shivering, so you send that information to the spinal cord. The spinal cord says, she's cold, we need to do something. The response in this case might be to cause those erector pili muscles to respond. Remember those erector pili muscles in the skin? What do? Raise the hairs. So especially if you're a mammal, with a lot of fur, like a polar bear, they're going to basically raise their fur to catch air to warm up their bodies. So that's how that's working. 3 separate nerves working together to take information and then cause a response. Okay, now I've put this in here just to show you that everything's not a simple loop. Okay, it's not a simple information in, integration, information out. There are some cases where you're going to have one neuron that may go and cause several different neurons to respond. Okay, almost like a cascade effect. So maybe a small stimulus will then cause many neurons to fire and cause a bigger response. So sometimes you're going to see this cascade effect. That's going to happen sometimes. You may see, again, where you have one neuron go and then affect 2, and then affect 2 more. Again, another cascade. These are divergent pathways. I just don't want you to get the idea that it's the same loop. It's not 3 neurons every time. There are different ways for these to work. Sometimes several different neurons will all affect the same neuron. If you need a response, you want a lot of information, you want a lot of stimuli, you have 3 different neurons, they all stimulate this one and you get a bigger response. This converging pathway can also be seen like when you're making a memory. So for example, maybe this input came from the nose, maybe this was smell, okay? And maybe this input came from the ear and you heard a certain sound. So all of a sudden you've smelled a rose and you've heard a train, and maybe you felt some vibration, and it reminds you of your grandmother's house next to the train tracks. Okay? That makes sense? You've got all this information coming in, and then your output is a memory that you remember. Oh, when I used to smell that smell and hear that sound and feel that vibration, I was at my grandmother's house. So it's all that information coming in, giving you a way to remember something. So there's a lot of different ways for your brain to work, lots of different pathways. So I don't want you to get the idea that it's just 1, 2, 3. Information in, information out. Let's see. Okay, let me